various principles and its related concepts involved in an emerging field called as biotechnology. So this might be useful for persons who are pursuing their undergraduate in biotechnology, and mostly this video is oriented towards people who are preparing for their competitive exams, especially NEET. And this chapter is given in NCERT class 12 syllabus. So this is a very important chapter, as we know that especially in the year 2021 almost some 5 to 6 or more than 5 questions were asked from this chapter. Though the reading of this chapter or in our textbook gives us an idea, but I believe this video, listening to this video lectures will help you to feel or really understand what these techniques are, why are they applied, all those things. So from this chapter, a series of biotechnology principles will continue in a sequence. So this video is mainly to explain what biotechnology is. So overall, this is going to be an introductory video for this chapter. So before we begin with this particular discussion, so you have to really know what a DNA is, what a gene is, and all those things. So please go back to my previous video in which I have clearly explained the differences between a DNA, gene, chromosomes in a video entitled "A Journey from Gene to Chromosome." Right. So that video is going to be the basic for understanding many of the chapters, and one among the chapters is biotechnology. So without those understanding the biotechnology chapter, trying to understand this chapter will be totally useless. Right. So coming to this, the word biotechnology. So what does it contain? So the very word biotechnology is going to explain what biotechnology is. So if I am going to dissect this word biotechnology, I can have two sensible words. One is bio, the other one is technology. So I hope we are quite familiar with both of these terms. That is the term bio as well as technology. So first, what is bio? Bio is something related to life. Right? Bio is something related to life. So here I mean a living organism. Okay? Living organism, which is where a living cell is going to be the basic unit of a living organism. On the other hand, you have the other term, technology. So what is technology? So technology, I did not teach you. What is it? So we are all in the world of technology. So technology means some technical advancements that we are going to apply in a living cell for a cause of betterment. Okay, so this is what is biotechnology. So we are going to apply various techniques. So before we move on to what are those techniques, what are those details, before we move on to all those details, I'd like to highlight few important applications of biotechnology, so with which we can understand these techniques in a very clear way. So we have a separate chapter called as biotechnology application, but I would like to highlight few important biotechnological applications. So biotechnology has got quite a lot of application in various fields in our everyday life, but we don't feel it, but it has got. For example, food industry, many genetically modified plants, we call it hybrid variety nowadays. This is a very common word that we hear, hybrid variety. So those are all nothing but the applications of biotechnology. So for many betterment, which we'll be dealing with a bit later. And in case of pharmaceutical industry, biotechnology play a very, very crucial role. How present day most of the Indians or most of the people in this world, especially the elders, due to whatever be the reason, lifestyle or pancreatic underproduction of insulin, so people are suffering from a major disease called as diabetes mellitus. We commonly call it as sugar. So diabetes mellitus, whatever it has got type 1, type 2. No, I'm not here to discuss all those details. But whatever be the disease, one final point or one final medicine to treat diabetes mellitus is artificial insulin. So you could have seen people having an insulin injection at their hand and trying to inject themselves, okay, so that their blood glucose level is maintained. So the question is, how do we get this insulin? So what sort of insulin are we using here? Yes, it is a human insulin. So many of us have a misconception it's a pig insulin is being used. No, it is a human insulin. From where do we get? Are we going to get from a human? No. I don't think so, because the very basic definition of hormone is they are produced in trace quantity. They are produced in very minute quantity. So obviously, something which is produced in very trace quantity or minute quantity, we cannot get it. Okay. So and moreover, the insulin stock what our pharmacies have in and around the city, okay, they are in abundance. So which means that the question, how are we going to get this insulin which is commercially used in present day? Then the answer is biotechnology. So through biotechnological process, okay, sufficient insulin hormone can be obtained. So how? So we will be discussing the various biotechnological techniques and its principle with the purpose of getting an artificial insulin hormone. So that would be the objective of this biotechnology series video, right? So our ultimate purpose is insulin hormone. So how are we going to get? So we are going to do something theoretically, experimental theoretically, okay? And at the end of this discussion, this series of discussion, we'll be able to see insulin hormone through imagination, right? So we are going to theoretically we are going to see this, right? So for this, what is this technology? I'll come back to here. What is this technology? This technology is what is called as genetic engineering. Right? Genetic engineering. So engineering means construction. So everyone know mechanical engineers, civil engineers. So they do some sort of construction work in their respective fields. So here we have got genetic engineering, and nowadays genetic engineering has become a subject in biotechnology in various universities, and very soon I expect a separate BTech, like BTech biotechnology, BTech genetic engineering could be a specialization in coming years. These are not so far to see them. So such an interesting discipline. So molecular biology, genetic engineering, these are all related fields with some minute differences. Right. So genetic engineering. So what is this genetic engineering? So engineering doesn't always means some constructing or producing something new, but it also means that altering something. We have an old house, we need to alter it, we call an engineer. So through engineering, various technology, engineering aspects, we are trying to do some alteration. So we got something new, right? So this is what is genetic engineering, and the principle of this genetic engineering is DNA1 plus DNA2 will give you rDNA. So this is the principle or the equation. And a word of caution, the word DNA here, what I have written or what I am trying to mean here, is not only the double-stranded helical structure, the structure which was proposed by Watson and Crick. So everyone would imagine that structure. So if I say DNA means double-stranded helical structure as proposed by Watson and Crick. So you could have seen this structure. Yes, this is DNA, no doubt about it. But in this chapter, biotechnology is something which is made up of nucleotides. So I call a gene as a DNA. Okay, something which is basically made up of nucleotide will be referred as a DNA, right? So this is a very general term we are going to use here, so you will not get confused. So DNA1 plus DNA2 will give me rDNA. So R stands for the recombinant DNA, right? Recombinant DNA. Recombinant. So combination is going to change here. So this is the equation. This is the principle. What is the purpose of? I said we are going to produce recombinant insulin hormone through biotechnology. So our purpose is going to be recombinant insulin hormone, for which I am going to take a gene here as DNA1, and I am going to take DNA2 as vector. Right. So what is my objective? Recombinant insulin hormone. So I should have which gene with me? I should have insulin gene. I should have insulin gene with me because I need insulin hormone. And I'm going to put it in a vector. What is vector? So I'll be having a separate discussion about what vector is. But for the sake of our understanding here, clarification, vector, it is a very general term. So your physics will suggest you something called as vector. Your mathematics has got something called as vector. In biology, there are vector, and I believe in two cases. So one is microbiology. That is a vector. What is a vector? An organism which carries another organism through which the disease is being transmitted. So the best example is Anopheles mosquito, which is going to carry Plasmodium, and disease is going to be the malaria. So we have some diseases called as vector-borne diseases, right? So here the vector is not such type of vector. Here the vector is molecular vector, right? The vector is molecular vector, which means that this is itself is going to be a DNA. It has to carry something. It will carry another DNA. So DNA2 is going to be vector, which is going to carry DNA1, right? So that's the very the basic meaning of the term vector. And there are lots and lots of examples of DNA to be called as vector, of which we are going to use here the plasmid. Right? We are going to use something called as plasmid as vector here. Getting the point? So I am repeating it. Vector are basically a DNA which will carry another DNA. So this is what is called as molecular vector, right? Which will carry another DNA. So among various vector, the most common vector, preferred vector is plasmid. So we are going to consider plasmid for our discussion. So how a plasmid will be? A plasmid will be generally a circular one. Generally, plasmids are circular, which are naturally found in bacteria. So if I am going to have my insulin gene, right? If I am going to assume that this is going to be the insulin gene, so I am going to insert this insulin gene into this plasmid. So now, before which I was having just the plasmid, now I have got a recombinant plasmid. Recombinant because there is a plasmid plus insulin gene, P plus I. So this is going to be the insulin gene and this is going to be the plasmid. So I can call this as a recombinant DNA. As I said, something is made of nucleotide will be called as a DNA. So you have to consider, always have that thought in mind. So DNA doesn't always denotes double-stranded helical structure. So this is a recombinant DNA, or to be specific, I can call this as a recombinant plasmid, or to be further specific, there is an insulin gene sitting there. So this is a recombinant insulin plasmid. Right? So we have got a recombinant insulin plasmid with us. Is it going to guarantee hormone? No. So this is just a gene. Right? We have to get the hormone out of it. So I write it here. This is a recombinant insulin plasmid. This is a plasmid in which we are going to insert the insulin gene. So now from this insulin gene, insulin hormone needs to be produced. So these are all further details to continue. So before that, the question that comes is, I said, we are going to, deal something theoretically, the practical aspects. So which means, let's say, how am I going to get this insulin gene? So what is the source? These are all the questions, that are rising here. So what is the source of this insulin gene? What is the source? And what is the source of this plasmid? So how am I going to get this plasmid, insulin gene? Right? So before I proceed with how do I get insulin gene, I would like to mention that the plasmid that I have here, I have two sources. One is, generally plasmids are extrachromosomal DNA found in bacteria. You would have heard about it in your Monera classes, bacteria. But either I can catch hold of a bacteria, I can get the plasmid out. This is one way I have. The other way is, go to a biotech company, get an artificially constructed plasmid which we call a synthetic one. So what do I prefer? So if you are going to ask yourself, which would you advise, whether to go a natural one, that is getting a plasmid from a bacteria, or getting from a synthetic one from a company? So I would always advise, or you yourself would advise, right, after listening these chapters, at the end of this chapter, that better go for an artificial from biotech company for various reasons. Okay, so this plasmid, we are going to get it artificially synthetic one from a biotech company which we have in various metro cities like Hyderabad, Bangalore, Chennai, right? So this is done. So plasmid, you can order it, you are going to get it. So how about this insulin gene? How that we are going to get this insulin gene? Just give a thought about it. Then you can visit my next video where you will get the answer. How do we get insulin gene? Right? Keep watching. Thank you.